Half a day and welcome to Freshman Genetics. In this video we're going to look at the sex-linked trait problems. Now in humans we have 23 pairs of chromosomes. 22, the first 22 are our autosomes, and chromosomes, the pair number 23 determines whether or not we will be a male or a female. And the presence of the X chromosome, if we have two X chromosomes, will be a female, and if we have an X and a Y, we will be a male. And this plays a role in certain genetic disorders, and that's what we'll be taking a look at. So the first thing we want to do is take a look at the basic Punnett Square for our sex. So how we end up with offspring is we have a girl and then we have a boy. So when the girl makes her eggs will always be X's. Okay, if we look at her genetic makeup, a girl is XX. So she must pass on an X to her offspring. However, we take a look at the boy and his genotype, and he has an X and a Y. So we know that some of his sperm are going to end up with the X trait, and some of his sperm are going to end up with the Y trait. So if we put this egg and this sperm together, we end up with a girl offspring. If we put these two together, we end up with a girl offspring. These two will be a boy, and these two will be a boy. So we can see here that roughly 50% of our offspring will be XX, which equals the girl phenotype, and 50% of our offspring will be a boy, or XY genotype, boy phenotype. And this has come true with most, if we look at large numbers of our population, we are roughly 50% female, 50% male. In individual families, of course, our statistics aren't as reliable with small numbers. And so you may find one family has a lot more girls or a lot more boys than others. But every new event, keep in mind our statistical rules, that every new event is a new shot, and so 50% of our offspring will be girls and 50% will be boys. And that is determined by which particular sperm is the one that fertilizes the egg. So it's the boy that actually determines the gender of the offspring. Unfortunately, that doesn't help any of those poor queens that were beheaded in England because they did not produce male offspring. It was actually the king's fault, but that's a whole other story for history class. So we'll move on. So here we have a human karyotype. First thing we'll notice is the 22 pairs of autosomes, which that is basically what makes up pretty much most of our genetics and pretty much all of the qualities of our humanness and then our unique qualities of our individual self. This last pair, the 23rd pair, we refer to as our sex chromosomes, and I wanted to pull this slide up so you could really see the difference between the X chromosome and the Y chromosome. So the X has a lot of information on it, and the male does not have two copies. He has an X and a Y. So traits that are on the X chromosome, he will not have a match for it. And that's what makes males more susceptible to sex-linked disorders because they have no way to be able to mask it with a normal gene. Whatever ends up on their X chromosome, that is the display they are going to have. That's what their phenotype is going to show. Now, females will have two copies of this X, so they will be able to possibly have a second shot at being able to be normal because they have that second copy that will hopefully be normal. And so it's a little bit more difficult for females to get the sex-linked traits. However, it is completely possible for them to still display these particular disorders. The Y chromosome mainly carries traits for maleness, and so a lot of the traits that we found on those are going to determine things relating to a male and his particular body parts and that type of thing. So those we tend not to look at this level of biology yet. All right, so now we're going to look at what genotype shortcuts we write in doing Punnett squares for sex-linked trait problems. So we're going to use the symbols capital D to indicate a normal condition and lowercase d to indicate a disorder. Now with our genotypes, it's very important that we include the X's and the Y's for these because that will make a difference as far as our offspring goes. So females we know have the combination XX. So she may have one of her chromosomes normal and the other chromosome normal. She may also have one of her chromosomes normal and the other chromosome has the trait for the disorder, has the allele for the disorder. And the other type would be X lowercase d, X lowercase d. And this would mean both of her X chromosomes have the disorder allele on them. For males, they can be X with a normal d and a Y, and X with a lowercase d and a Y. So we notice with males there's no superscript on the Y because the Y chromosome does not affect our phenotypes at all. So the males can either be normal or have the disorder. There is no in-between for them. Females can be completely normal in the case of this individual. This individual, when they're heterozygous, they have one copy normal, one copy with the disorder, are called female normal carriers. And that's because they're going to display the normal characteristics, but they're going to carry this disorder and can pass it on to future offspring, or it can be passed on down to future offspring or grand offspring and great-grand offspring. It can end up somewhere further down family tree. And then you have females that have two copies, and they are going to display the disorder, whatever that may be. So in the problems we're going to look at, we have some common sex-linked disorders that we are going to use in our problem sets. So the three most common that tend to be used in freshman biology class are colorblindness, hemophilia, and muscular dystrophy. And some of you may have heard of these disorders previously. Colorblindness is when the individual has difficulty differentiating between some colors. Hemophilia is a blood disorder in which the person has difficulty in clotting their blood. And muscular dystrophy is a condition in which the muscles will dystrophy at a certain period of time. And of course scientists are looking at all of these disorders to find ways to be able to cure them. But for right now we're just going to look at the basic genetics behind them. So the first problem we're going to look at is a colorblind male and a normal female. Now the normal female is not a carrier, okay? So we're going to put NC, which means she's not a carrier for that. So first we'll determine the genotypes of our two parents. So the colorblind male, he will be male, so he has an X and a Y. He's colorblind, which means his X will have the lowercase d. The female will be XX, and she is not a carrier, so she will be capital D, so she'll be completely normal. So if we place our male here, we have X lowercase d that he may pass on, or the Y. Then the female will be X capital D and X capital D. So if we look here and complete our squares, bringing our X's down and our other X's across, and then our X's down and our Y across, we can see that we end up with this. Both of these will be females, and they will both have one capital D, one lowercase d. So their females will be normal, but they will be carriers of the disorder, which in this case is colorblindness. So we have two females, potential offspring, normal female carriers of colorblindness. And our males, we look at the males and we see that both of those will have the regular normal trait. So both males will be completely normal males. They will be able to see fine. And we can look to see that's because they obtained the Y from their father, and they obtained their capital D, their normalcy, from their mom. And the dad, all of his female offspring, he passes on their X chromosome, so they will all end up with that colorblind trait that they may pass on to their offspring in the future, but they themselves will be normal. Now if we take a look at a man who has muscular dystrophy and a female carrier. So the man will be XY, and he has the disorder, so it'll be lowercase d. The female is a carrier, so she will have her two X's. One of her X's will be for normal, and the other one will be a lowercase d, meaning she has the disease. So again, we can set up our Punnett square. And for the female, we have the X with the capital D, X with the lowercase d. For the male, we have the X with the lowercase d and the Y, and we put these together just like we did previously to find out our potential offspring. So we have here, they get these traits possibly from the parents, and this is just looking at the potential for the offspring that we see here. All right, so looking at this, these are our female offspring, and of the females, this female would be a normal individual with a carrier, so she would be a carrier for muscular dystrophy. This female would have the disorder. She has both of the lowercase d's, so she would have muscular dystrophy. This individual would be male, and he would be completely normal. And this individual would be male with the disorder. So if we look here, if this particular cross were to happen, a male with muscular dystrophy and a female carrier, we notice that about 50% of their offspring would have the disorder or would potentially have the disorder muscular dystrophy. The other 50% would be normal. However, all of their female children would carry that disorder, which could be passed on to future generations. So these types of crosses may influence whether or not a man and a woman want to produce biological offspring, or this may be a case that if they understood what was happening, they may opt to come up with maybe adopting children or doing something else. Basically, that's what these crosses are going to look at. What is the chance, the probability? And they have researchers now and geneticists that if they know that there's some type of disorder in a family, they will go to that person and they will be able to find out much more information than we can talk about on this short video. All right, so now we're going to look at a normal male with a female with hemophilia. So what I'd like you to do right now is to pause the video and see if you can fill out and come up with the ratios that will happen in this particular cross. So just hit pause and do this out on your Punnett square on a piece of paper and see if you're able to come up with the right answer. So here we are looking at our potential offspring. So our male, our normal male, is XY, and he has the capital D because he's normal. The female is XX. She has the disorder, so we know she must have two lowercase d's. We put our Punnett square here, and we have the X capital DY, X lowercase d, X lowercase d. Looking at our offspring, these are our female offspring, and both of our female offspring will be carriers for hemophilia, but they will display a normal phenotype. And here we have both of our males, and our male offspring, XY, will both have the disorder because they will both have the mother would have passed that gene on to them. So if we were to just look at our children, all females would be carriers, and all of our males would be afflicted with hemophilia. I hope that this video helps you, and have a great day.